thank you everyone for joining. Those, if people are coming in, that's fine. Just keep on coming. Just to maybe give a high level intro to where we're at. The work that we're going to present today in this webinar is a culmination of almost 4 years of work by the Sentry team. We have over 180 wastewater treatment facilities globally that we monitor. We're monitoring the influent wastewater primarily, that influent wastewater quality. We're measuring surrogates for BOD or rbCOD and the biomass health. And over the last 4 years, we've learned an awful lot about what causes process upset. And specifically today, we're going to talk about EBPR and really focusing in on carbon and why carbon is so important and the conditions around EBPR and how carbon is so central to that. So that's where we're going to get to. Carbon management is key, or my favorite phrase, got carbon. Thank you, Sergio. So here we go. Okay, so the two presenters for today, Juliana Maya-Franco. So Juliana is the Director of Product Development at Sentry. She's also a part-time professor at Algonquin College in Ontario in Canada. She's an IWA Young Water professional. And most importantly, she's an arts, cats, and travel lover. Thank you, Juliana. And we're also being joined by Sergio from Ovivo. Sergio is the Process Aeration Group Manager for Ovivo. Remarkable track record at delivering solutions, treatment solutions, not just in the US, but in Italy, Spain, and Chile. Has a passion for bugs and percussion, and central to the Jamming for Water. So if you guys want to see more of Sergio in person, then make sure to sign up for Jamming for Water at WWTPs. Okay, so let's get started. The quick agenda. So what we're going to start with, Juliana is going to take us off and just really high level discuss the key conditions for a bio-P. How can you create good conditions for reliable bio-P? But then also touch base on those challenges. Like we know wastewater is variable, and how does that impact those when we're trying to create those reliable conditions for PAOs and bio-P? With that as a base, we'll quickly just introduce what the Sentry technology is, how we can measure rbCOD biomass health in wastewater in situ. We then have 4 case studies. The first 3 we'll go through reasonably fast. Case studies from Germany, Canada, and the US. And then we'll hand it over to Sergio to talk in more detail related to Idaho Falls and their work with Sentry, assessing this tool as a new lens for the EBPR process. Okay, so with that in mind, Juliana, will you take us away? And let's just start with that intro to bio-P and let's discuss the challenges associated with EBPR in general.
Perfect. Thanks, Pat. Yeah, to kick off the webinar, I'd like to do a quick introduction to biological phosphorus removal. That way everyone is on the same page, okay? So to get rid of phosphorus that is dissolved in the water matrix biologically, we rely on living organisms that capture that phosphorus. These organisms are called phosphorus accumulating organisms, surprisingly, or PAOs, and they look like this in the image and to the right of that slide. So the dark spot that is labeled as PP, it's a granule of poly-P or a storage of phosphorus inside one of those cells. In that image, you can also see something called PHA, which stands for polyhydroxyalkanoate. And that is a polymer that works as a food reserve that enables this phosphorus accumulation. So in short, you need this PHA in order to have phosphorus accumulation later. So, well, this PHA is important. Sorry, Pat, could you go back? I'll explain the diagram to the left. So how is that PHA get to that cell? That fuel? Well, under anaerobic conditions, the PAOs uptake short-chain molecules of carbon or volatile fatty acids and convert them into PHA. So that cannot be done for free. They need to use some glycogen as a store that is in the cell, and also release some phosphorus, like some poly-P, as a source of energy. So this starts being a bit counterintuitive because for this process to work, you need first to release some phosphorus, create this fuel as PHA, and then be able to uptake the phosphorus. Okay, so the next one. Okay, so this all makes sense because in aerobic conditions, the process is reversed. So instead of releasing phosphorus, now the cell uptakes it and accumulates it in excess, leading to this net removal of phosphorus that we all want. So in short, we want three main conditions for the EBPR or enhanced biological phosphorus removal process to work. We need first that anaerobic condition without any other electron acceptors, such as oxygen or nitrate, just very, very anaerobic. Then you need also a VFA concentration that enables this fuel, let's say, build up. And then the switch from anaerobic to aerobic so that the cell can start uptaking that excess phosphorus and remove it from the solution, okay? So I think that that was a very high-level explanation of how EBPR works. Let's see what are the challenges for this process. So we might have some control over how aerobic or anaerobic an environment is. That's a good job for engineers and operators. But we don't have that much control over how much VFA enters a plant or how readily biodegradable that carbon is in the plant. So we know that wastewater is very unstable. We certainly know that carbon, the nature of that carbon is always changing. So this composition of wastewater usually shows a diurnal pattern that's already very dynamic. Now consider adding extra variability to that dynamism by having some rain infiltration or, let's say, industrial contribution from the collection system. And that's only for composition. Now, how available that carbon is for the biomass in a plant also can change from environmental factors such as seasonal temperature changes or the presence of inhibitory compounds. And without that information, is really little what you can do for EBPR. So here I'm showing in this graph an example of biological information that we can provide that tells you how much carbon is actually available for biomass. You'll see rbCOD as how we report it, readily biodegradable chemical oxygen demand, and that's the profile of available carbon in a plant in Germany. So you'll see that there is a diurnal profile, very clear, but then during the weekends, that profile kind of distorts, and that's exactly when the plant starts reporting issues at their effluent, so some phosphorus exceedances. So the message here is basically that if we know the right information and time, we have better control on EBPR, okay? So, well, I'm not saying that plants cannot measure carbon. I mean, plants have many different ways to measure carbon, like COD, BOD, TOC. But the challenge here is that these metrics are chemical and they don't usually give a complete picture of what fraction of that carbon that is in the water is actually useful for the EBPR process. So as I mentioned, operators can use these chemical metrics like COD, BOD, TOC, VFA to quantify the concentration of organic matter, but those metrics won't consider factors like changes in temperature or presence of inhibitory compounds, or even how readily biodegradable the carbon is, which at the end will affect the availability of that carbon to the biomass or to the PAOs. So in contrast, we have biological sensors, or there is this biological option to monitor the carbon that will give you the information and fill that gap in this process, okay? So is this new lens, this new biological insight on carbon availability for EBPR, what operators lack to have better control of their process? Because they really know where the sources of carbon are, like specifically for VFA. If the influent doesn't come with enough VFA for EBPR, they can resort to RAS or primary sludge fermentation to get some VFA. And if that's still insufficient, they can also dose some supplemental carbon. So they can play with what they have, but the key is to have the right information at the right time to enable these optimization strategies.
I love that, Juliana. And that's exactly the key point is we know that we can get carbon from different parts of the process, but is it the right carbon? is like BOD necessarily isn't the right carbon, it's BOD5. Like if that carbon isn't bioavailable for your PAOs in the anaerobic selector, is it actually helping you or not? So having that tool, that lens that allows you to measure what matters is really important if you're trying to manage that carbon and maintain those conditions for PAOs, those conditions that they really need to succeed. Okay.
Yeah.
Thank you. Okay, so we've summarized at a high level what is BioP, EBPR, what are the conditions from a biological perspective that we want to maintain to select for those PAOs in an anaerobic environment, in an anaerobic selector environment, and that potentially if we had data or a signal that could tell us exactly that food, that available component, that we could use this to better understand carbon management and better maintain those conditions for reliable EBPR. So next, what we'll do is we'll just really quickly touch base on the Sentry sensor and how that works to try and link the two together. And what I will say as well to folks is if you have questions, just write those questions in the chat, and we can get to those at the end. So do, as the conversation's going on, as the webinar is going through, feel free just to plug in your questions there, and we can get to them at the end. Okay, Juliana, how does Sentry work?
Yeah, thanks. So the Sentry monitoring platform uses something called bioelectrochemical sensor. That's a big word. It only means that it's a sensor that allows naturally occurring bacteria from the wastewater in the process to grow in the electrode, thanks to some structure like tiny micropilli that attaches to conductive surfaces in the sensor. So you'll see in that background image behind the sensor, that's a picture of part of a bacteria that we inoculate in the sensors. And these little strands are little hairs that can connect to the conductive surface on the sensor. So as that bacteria consumes organic matter from the water matrix, the sensor converts that into a digital signal. So in a very real sense, you get to see or view how much food is available for the biomass in the plant and measure that as a digital output. So I bet most of you must be wondering, how is this even possible using bacteria? So in a more technical explanation is here. So the sensor has an electrode. You'll see the two plates of an electrode, anode and cathode there. And we grow that biofilm in the anode side of it. We also apply a voltage in the circuit so that as the biofilm degrades carbon biologically, all the resulting electrons from that redox reaction are respired to the electrode and not to other electron acceptors. So oxygen or nitrate is not involved here. So everything is going to the anode. And this flow of electrons, which in physics we describe as current, is what we measure. So we measure this current. And this current metric is directly proportional to the amount of food or biodegradable or readily biodegradable carbon that is available to the biomass in the system. So in other words, what we're measuring is basically the ability of the biology to process or to treat the wastewater, or in layman terms, we like to say that this is a heart rate monitor for the biology. And well, this measurement is translated into a minute-by-minute data set and that represents the amount of organic material that bacteria in a system can consume. So we report this as consumable VOD or consumable rbCOD or VFA, depending on where the sensor is installed. And that word consumable is what actually tells you that this metric is not a concentration. This is what is actually available for the PAOs in the plant, what is consumable, okay? And this is displayed in a user-friendly platform. You'll see there like a screen, but it could also be integrated directly to a SCADA system in a plant. Okay. So I think that next is case studies.
Yeah, okay, love it. So as I mentioned at the beginning, we do work globally. We have over 180 clients globally, and we've grown this base of clients where we're monitoring their influent wastewater conditions over the last 4 years. And today, what we're just going to do is focus on a subset of those. And it's these ones where EBPR has been a challenge or has been identified as a challenge. What I would also say is routinely when we hear from potential clients, I have a challenge with my EBPR process. Invariably, the logic or a lot of the feedback we get initially is we think we suspected some toxicity from an industrial client. That tends to be from an operator perspective, the go-to rationale for why we're having these upset events. And what I would say is a lot of the time that isn't the case. In terms of the tool and how it's installed, just to give one image of the Sentry sensor installed in a process, we just want the sensor in wastewater, in the flow of that influent wastewater, typically post bar screen, just before your primary clarifier, perhaps at the effluent of a primary clarifier, or perhaps in your anaerobic selector. And it is designed to be very simple. It's designed to be just a surface that has the biofilm from your process growing on the surface. We don't wish for that surface to be cleaned regularly. Yeah, no chemicals, no moving parts, nothing like this. So it's just a surface where the biofilm is continually consuming that carbon, that rbCOD in your wastewater. Okay, so for today, Juliana, do you want to take it away? So we're going to do Summerside, Clean Water Services, and Neumarkt in Germany. Love it.
Awesome. Yeah, thanks. So yeah, the first one is Summerside in our home province, Prince Edward Island in Canada. That's the paradise for lobster lovers, if you don't know what PEI is. So this is a small, or let's say mid-sized municipality, and they treat municipal wastewater, but as well some dairy industry effluent. And they came to us because they were having issues sometimes with phosphorus and nitrate breakthroughs. So they were thinking that it could have been some toxicity issues in their influent. They just needed to know some more information about their, say, carbon profile. And we installed sensors in their influent after screening. After some months, we found that there was a big deviation of this consumable BOD portion during some days, especially when the dairy industry shuts down. And that's exactly when they started having phosphorus and nitrate breakthroughs. So in these two images, you'll see that very clearly. So when the consumable BOD signal from Sentry went down below a certain threshold, they started having issues with nitrate in their effluent and also phosphate in their effluent. So it was not a toxicity issue, but more like a nutrient, carbon deficiency issue in there. And with this information, they were able to first have some more conversations with the industry in the city and like plan this better so that the exceedances were not happening that often.
Love it. And I'd say, Juliana, this was the first time, this was maybe 4 years ago, that we began to understand there are those certain thresholds, even at the influent, that if we can just keep the signal above those thresholds, your BNR process is fine. But as soon as that signal goes below those thresholds, you do tend to have problems. Okay, number 2.
Yeah, so number 2 is very similar. It's in Germany, in Bavaria region, and a very similar size, let's say, treatment plant, also treating municipal wastewater and also treating some industrial contribution, but now from the brewery industry and also some hospitals. They came to us again with similar issue. We're having some phosphorus breakthrough, and we don't know exactly what that is. It could be toxicity, it could be something else. So we installed sensors in their influent as well, just following what we had learned from Summerside. And we also saw that deviation in their consumable BOD portion during some days of the week, especially during the weekends. So this is the same image that I was using some slides before, but this is to show you that, okay, during the week, we could see that journal pattern, and then during the weekends, that signal went down below a certain threshold. That's when they had this phosphorus breakthrough. So we went and had this next step on, hey, could you do something with this information? Could you start adding some supplemental carbon to this process during these moments and see what happens? So the next week, they added some glycerin. There is a dose there. And they saw that it really aided the phosphorus removal process. They didn't have that exceedances, like the exceedance issue. And that was basically a validation on how signal or the Sentry signal was not just to collect information and understand the profile, but also was actionable, okay?
Yeah, I love that because that brings us right back to the start. You're measuring what matters. We know there's enough carbon, whether it's from the influent or whether we need to get some from a RAS stream, fermented RAS, or the third option then is that supplemental carbon. And it's, can we just maintain these carbon thresholds using one of those three to maintain it, keep it above a certain low threshold? And if we can, we know we'll be okay for our BNR or our EBPR. Okay, so number 3, slightly different.
Yeah, number 3 is slightly different, as Pat says. So we were working with Clean Water Services, the plant at in Tigard, Oregon. This is a much larger facility, and they also treat municipal wastewater. But their issue is that they have very stringent seasonal nutrient limits. Specifically for total phosphorus, they have 0.1 milligrams per liter, which is very restrictive. So they were piloting different processes, RAS fermentation processes, and testing sizes in their aeration basins. So we installed sensors, not in the, well, also, but not for this specific purpose. We installed sensors in their anaerobic selector carbon, let's say, basins. And we had 2 trains to monitor. So one that was larger train, larger anaerobic basin, the other one was smaller. In the larger one, they were also testing RAS fermentation. So the idea here was to understand what was the profile of BFA in that RAS fermentation and have some comparison between the 2, let's say, basins, understand if there was any relationship with phosphorus as well. We were installed right next to orthophosphate analyzers as well. So that enabled us to do a correlation with that, let's say, metric, orthophosphate concentration. And we found in both basins, the larger and the smaller one, that the correlation between the Sentry signal, which is readily biodegradable organic carbon, or VFA, was very, very close to orthophosphate. And all that makes sense. I mean, if you have higher phosphorus, which is higher orthophosphate release, it means that you had higher VFA availability in the water. So that was basically the validation on how we could interpret this signal in anaerobic basins. And this helped us also understand that this could be a better location for this kind of monitoring. So this could be analogous to orthophosphate. Orthophosphate sensors are very, let's say, complicated to use and time demanding and costly as well. So if we could have this other option, it would be better. So with this in mind, we basically decided to go forward with another project now in Idaho Falls. So, Bud, could you go next? In Idaho Falls, now having learned all that, where to have the sensors, how to interpret the signal, and we wanted to basically understand if that relationship with orthophosphate and BFA stayed in the, well, like was still consistent in this plant, and have better information for integration into a new process. So I'll let Sergio elaborate on this.
That's a
Hi,
perfect transition. Okay, so thank you very much, Juliana. So we've been through basically 3 case studies on our side, and then that led us up to this work with Sergio. So Sergio, let me lead it to you. You take it away, and let's go for it. Idaho Falls.
All right. Thank you, Patrick, and good day to everybody. It's a pleasure to be here. Patrick invited me to co-present and share a little bit about our experience with the Sentry probe at Idaho Falls. You can see my contact information. I've been doing biological treatment for 23 years. Bugs is one of my passions. The second one is percussion. You can see I play during WebTech for Jamming for Water with other water professionals, which has been a great enjoyment for me. Anyway, originally, right, Ovivo and Sentry have been talking for probably the last 2 years. And the main reason for Ovivo, it is a new development within the carousel product line that we introduced to the industry this year. We took our well-established 3-stage carousel system with anaerobic, anoxic, and the ration zone with that typical racetrack, as you can see on the screen here, for the oxidation ditch. So we took this well-established configuration and did a big upgrade, big development upfront, looking at the anaerobic and anoxic selectors, understanding that the driving force upfront is carbon, more specifically soluble carbon, as we have seen so far in this presentation. And our main goal with this new configuration is two things. Number one, maximize the availability of soluble carbon upfront and make sure that it's utilized upfront. We want to avoid bleeding soluble carbon into the aeration basin. And the new concept is carbon management, looking at all the sources that we have upfront that we can tap into to generate this soluble carbon and, most importantly, VFAs, right, to grow PAOs. Next slide, please. So with that in mind, we introduce what we call carousel selector. It is, again, an upgraded version of the well-established three-stage carousel system with primary focus on carbon. Next slide, please. So why carbon? Number one, we all know carbon is the engine for BNR treatment, right? BioP and denitrification heavily rely on that soluble biodegradable carbon. As Juliana just said, BioP is more specific. We need to produce VFAs for PAOs to do the cycle and the wonderful thing that they do with BioP. The second fundamental element here that we introduce with carousel selector is with carbon management, right? Is understanding that carbon is a driving force for good settling sludge. So in our new development, right, we want to create a feast phase. All you can eat, all you can store in this case. We want to promote the growth of carbon storing bacteria. PAOs is one of them, right? And we know that if we have enough population of PAOs in the total mixed liquor, we will gradually start changing the morphology of that mixed liquor towards a densified mixed liquor with some insight of aerobic granular sludge. So we have seen plants, when you have enough of this carbon storing bacteria, you start seeing maybe 50% of the mixed liquor composed of particle size between 200 and 400 microns. So that the small granules that really can help you with more robust BNR treatment and having a good secondary clarified effluent and potentially even more capacity as you improve that settling, right? Next, please. So going back to Sentry, right? Why this is important for us? For us, became a new lens to see carbon. All this is about carbon with this development. The standard practice today in the treatment plan is to run the biochemical oxygen demand, right, test to understand your organic load. That's the BOD 5. There is a 5 there because it takes 5 days to cultivate this in the lab. So you take your sample, a BOD sample on Monday, most likely you will get the result a week after that. How do you use that number? Only for reporting purposes. You have nothing to do with the operation of the plant or improvement of performance because it's a week late already. With this, the intent with the Sentry probe is to have real-time monitoring that soluble carbon coming in to understand the dynamics, the unit profile, and from there, that visibility allows us to implement operating strategies with some tools that we have implemented in carousel selector. The second element for us was important about Sentry. It is a very simple probe. Simplicity is fundamental for carousel. Our clients love the simplicity. I can tell you that there is no need to calibrate this probe. There is no need to change membranes or light, anything. The only thing that we learn is that maybe once a month, every other month, you pull up the probe from the basin and just clean it a bit that excess biofilm, right? Very simple, perfect marriage with carousel. Next, please. So we have been working with this treatment plant, Idaho Falls, rated for 18 MGD. They have very interesting elements. We have been doing research with them for the last 3 years. And for this particular trial with Sentry, right, we want to correlate the Sentry signal in the influence of the bioreactor with other carbon metrics like BOD or the flocculated filter COD, right, the truly soluble carbon. So as you can see in this graph here, in this image, we put the first probe in the primary effluent. So there is a primary clarifier. So most of the BOD get into the first cells of the bioreactor soluble BOD. We put a probe there, but then we decided, hey, let's put more probes through these anaerobic selectors that they have in series to see how that gradient looks like, right? But today I want to focus strictly on that primary effluent. Primary effluent, Sentry signal versus BOD and FFCOD. The plan is very simple, right? You have those anaerobic selectors, great performance in BNR, PAOs, live population. They even have small granules. After those 3 anaerobic selectors, they go through a channel, as you can see here, and then split the flow between 3 trains in parallel, starting with swim zones, diffuse aeration, clarifiers, filtration. Next, please. Instead of the installation, very easy for us. Between one person from Ovivo, one person from Sentry, it took less than a morning, right, to install 4 probes, 2 panels, and all the electrical, conduit and things like that, and plus the safety elements. So from that perspective, super simple to install, cleaning, very simple, once a month, every other month, you pull it out, clean it a little bit, the biofilm, and you're good. Next, please. All right, first set of data. So this is data from the end of October through mid-March, so about 5 months. In orange, you can see the daily composite BOD that the plant does in-house, right? And in blue, you can see the Sentry signal. That is the daily average of all the data points that we take per day. This probe takes every minute a signal. So each blue dot is the average of more than 1,000 data points. As you can see here, it was the main things, aha moment for us. There is a strong correlation between the Sentry signal and the composite BOD from the plant, right? We were not intending to measure BOD per se, but the key for us is to understand those dynamics on real time, right? Remember, BOD takes 5 days. This is on time. One lesson learned here I would like to share is that if you want to correlate the signal to an actual BOD measurement, BOD 5 is done at the lab at 20 degrees C. Well, guess what? The influence to the bioreactor is not at 20 degrees C. So we learned that we had to do a temperature correction to normalize it to 20 degrees C. I had to give credit to the Sentry people to help us with that. And even I'm very happy that they're working on a new development to introduce the temperature signal into the probe and do the auto correction or temperature correction behind the scenes. But again, for us, that was an aha moment to say, hey, this really is an accurate visibility of the carbon coming in, right, to this plant. Next slide, please. We went ahead and did some flocculated filter COD in a lab. We had to pay extra for a little bit, plus the BOD and the Sentry signal. As you can see, again, great correlation between those 3 data. Absolutely, that's exactly what we're looking for, right? Have that visibility and make sure that the Sentry signal is accurately, right, following the dynamics of the soluble carbon coming into the plant. Next slide here, please. This for me is the game-changing aspect of Sentry. It gives me his 2 weeks the diurnal profile of the influent carbon. It's a new tool in the toolbox. We didn't have that visibility. That's what we call it, a new lens to see carbon. You can clearly see these 2 weeks, right? The peak loads, the low moment, probably at night. And with that visibility now, I can implement certain strategies with new tools that we have implemented in carbon soil selector in those anaerobic and oxy-basing to do the things I mentioned before. Drive better, more consistent BNR treatment, and try to drive this carbon storing bacteria population in the system to enhance cellularity. Next, please. So applications of the Sentry probe in carbon soil selector. This is how we see the landscape moving forward is being incorporated in selector. Number 1, we can optimize the food to microorganism ratio in the first anaerobic cell. To drive this carbon storing bacteria, we want to avoid diluting the carbon coming in. So if I have the carbon signal from Sentry, it's going up, I can send more RAS to that first anaerobic cell. If the carbon signal is coming down, I can send less RAS to that first anaerobic cell and maybe bypass some of the RAS, right? The key is that in the first anaerobic cell, I want to maintain a high, sustained FM ratio. Number 2, in terms of applications, I can optimize the internal recycle flow, nitrate. So more nitrates I can send up front if I have more carbon. So the carbon goes up, the carbon signal, I can bring more nitrate. With carousel, we had the benefit that we don't need pumps. We just use a gate and take advantage of the hard circulation pattern, right, the racetrack, and we can bring as much nitrate as we want, depending on the carbon signal. So a strong correlation between carbon signal and our ability to denitrify in this case. Next, please. The third element, this is a pretty cool one, I like it, is implementation of a fermentation cycle within the anaerobic and oxy-basins. We have a very innovative way to do that. And with fermentation, again, carbon management, we're looking at where we can tap into to produce these volatile fatty acids, right? And with fermentation, we can tap into the complex organics, the slowly biodegradable carbon, going through hydrolysis, fermentation, and increase that VFA quantity for PAOs. To do that, we now know the carbon profile and we can optimize the fermentation cycle. And in terms of this carbon, storing bacteria, we can maximize the feast phase, right? All you can store, all you can eat pretty much, buffet for PAOs up front, right? And it's pretty cool to see the carbon gradient, the soluble carbon gradient through the anaerobic and anaerobic selectors. As I said before, we want to avoid bleeding that precious soluble carbon into the aeration basin. That doesn't do anything for us. I want to make sure that that soluble carbon is utilized up front, and this can give us that visibility. Next one, please. With that, I did not have time to explain you more about carbon soil selector. You have my contact info. Feel free to shoot me an email. But for us, the key here was, number one, the simplicity of the probe, give us a good, accurate reading of the carbon. The next step in terms of the trials would be to take this to Florida, warmer weather, more weak BODs, and probably next year I'll be sharing some data of that new trial. But we are very confident that this is, again, the new lens to see carbon and start driving better processes, right, for BNR and densify mixed liquid. And that's what I have to share today.
If anyone has any follow-up questions, or would like to discuss potential projects, yeah, love it, Mark. That's exactly. You can reach out to Sergio on the Ovivo side and the Carousel side, or yeah, you can come directly to myself or Juliana. Our email contacts are at the start of this presentation, so I'll put them up on the slide just before we go. And yeah, you can reach out to us by email or reach us through our website. Yeah, we look forward to supporting you guys. I'll just quickly see if there's any more questions. No, I think we're good. Mark is good. Okay. On the button, folks, 45 minutes. Sergio, thank you very much. That was absolutely fantastic. This is really great. Juliana, thank you for everything. You're awesome. And yeah, folks, just reach out if you have any questions, comments. Feel free to reach out to myself, Sergio, or Juliana. Thank you very much.
Thank you.
Thank you.